Hello student, in last lecture we talking about the acquired immunity, its properties and the components like T cells, B cells, complement system, APC and the cytokines. Also we see the what is meant by the active immunity, passive immunity and their differences. In this lecture we talking about in detail of the active immunity, how it produces in the body of organism and how it resists the microorganism to get invaded in body of organism and proliferate. So basically, the active immunity is resistance developed by an individual towards an antigenic stimulus. Whenever the microorganism get invaded in body of organism, then the active immunity, that is adaptive immunity, start to get response against those microorganisms by producing the antibodies. So active immunity may induce by naturally or artificially. First of all, that is natural active immunity. For example, measles. In Marathi, we know that the kanznia. So measles virus infection occur naturally, and we get naturally immunity against those measles virus. After first exposure, the second infection doesn't carry even after the exposure of measles virus to the organism, because we produce the active immunity against those measles virus. And secondly, that is artificial active immunity by producing the measles vaccination. We know that the vaccination consists the half killed all killed viruses or bacteria. So these vaccine are ingested in the body of organism, which is essential to produce the active immunity against the measles. That is two types. So active immunity having two types, that's natural active immunity and second one, that is artificial active immunity. So basically natural active immunity are produced in the host immune system, which fight against the microorganism, bacteria, viruses, pathogen and any other foreign material. So active immunity is long lasting. So active immunity usually lasts for the longer period, but the duration varies depending on the type of pathogen. So basically this active immunity duration is depending on the type of pathogen. It is maybe long lasting or last for this short period. For example, the last lifelong following the certain viral infections such as chickenpox, measles, smallpox, mumps, rubella. We know that the chickenpox, measles, smallpox vaccination are given to the small childs, and these vaccination are useful to produce the active immunity in the body of fetus or children. So whenever the organism or human having the exposure or get exposed to the measles or these viruses, then they start to get response against those microorganism or viruses. That is active immunity having the last lifelong activity or active in the body of organism, but in some kind it is having the short period for the some infectious viruses like influenza. So they are active but in short period. Also this immunity having the pre-immunization and concomitant immunity. Immunity may last as long as the microbe is present. So basically active immunity is present as long as microbe present in the body of organism. Once the disease is cured, then the active immunity is stable and get their side, but the patient becomes susceptible to microbe again, because active immunity are active till the microbes present in the body of organism. When they eliminate the microbes from the body of organism, active immunity get silent, so that patient or that organism may become susceptible to the microbes again. So we can get the exposure of microorganism and form the disease in the body of organism, such as a plasmodium. So the immunity may last as long as the microbes is present, and once the disease is cured, the patient becomes susceptible to microbes again. So active immunity may not be protective at all. So basically this active immunity sometimes may not be active or protective at all in the some conditions of the organisms. For example, the Haemophilus ducreae. So the patient may develop genital lesion following the reinfection even while the original infection is active. So in active immunity, in some cases, this active immunity are not useful for the some kind of infection such as a hemophilus decrease. So this infection following the lesions by forming the genital lesion by following the reinfection, while the original infection is also active, that not be cured at all. So sometimes active immunity may not be protective at all. So there are two type of response given by the active immunity, that is primary immune response and secondary immune response. So first of all, that is primary immune response. When the antigenic exposure occur for the first time, the following event take place. So basically, whenever the exposure, antigenic exposure take place at the first time, some events take place. That is first one, that is latent or lag phase. That is active immunity develop which correspond to the time required for the host immune apparatus to become active. So latent or lag phase means the following the first exposure to the foreign antigen or antibody, and lag phase occur in which the no antibody is produced but activate the B cells are differentiating into plasma cells, and the lag phase can be as 2-3 days but often is longer, sometimes as long as weeks or months. So latent phase essential because in active immunity cells develop the B cells or T cells response against this microorganism, that is primary immune response, which producing having the time required for the host immune apparatus to become active like B cells, T cells or other cells, and this time period is called as lag period. Secondly, that is effector cells. So majority of activated T cells and B cells against the antigenic stimulus become effector T cells and B cells. We know that the acquired immunity is consist the mediators like T cells and B cells. These T cells and B cells are useful for the initiation of adaptive immunity against the microorganism, where T cells is one of the major component of the adaptive immune system, which having the role including the directly killing, that is T killer cell, or which is also called as cytotoxic T cells. These T cell producing the cytokines and regulating the immune response. So T cells promote the killing of the cells that have ingested microorganism and present foreign antigen on their surface. Another class of T cells called as regulatory T cells function to immune response and resolve inflammation. So effector T cells such as the helper T cell and cytotoxic T cell. So effector T cells like helper cell help to B cell to activate the production of antibodies, where cytotoxic T cell produce these cytokines or chemokines which is initiate the response against the microorganism. Also, some effector B cells also include the plasma cells, where B cells are the B lymphocytes, which are the type of white blood cell of the lymphatic subtype. Their function may be the humoral immunity component of the adaptive immune system by secreting the antibody. So main functions are to make the antibody against the antigen, to perform the role of the antigen presenting cells, and to develop into the memory B cells after activation of antigen interaction. So basically these effector B cells also include the plasma cells which produce the antibody against the microorganism. So in a primary immune response, they also having the memory cells. We know that the T cells and B cells are the memory cells. A minor proportion of stimulated T cells and B cells become a memory cells, which are the key cells for secondary immune response. Whenever the microorganism get invaded in body system in second time, with T cells and B cells having the memory for the recognition of that microorganism, and they recognize them and producing the specific antibodies immediately and kill them. Next one, that is antibody search. So and in antibody search, the activated B cells produce antibodies, specifically immunoglobulin M type. So in primary immune response, Activated B cells produce antibodies, mainly immunoglobulin M type, in primary immune response. So antibody appear in the serum in slow, sluggish manner in primary immune response, because this process is very slow and sluggish manner, because it is the first time antigen are get invented in body of organism, so that the adaptive immune system have to recognize that microorganism, that antigen, so that they can produce the specific antibody against that microorganism, bacteria or viruses. Then slowly they pick and maintain the level for the while, and then fall down. Then finally the low titer of the baseline antibodies may be maintained in the serum. So suddenly they pick the uh, production of antibodies, maintain the level for the while, and then fall down. And finally the low titer of the baseline antibodies may be maintained in the serum for the response against the microorganism which proliferate in the body of organism. So this is the figure in which we can see that this is the lag phase, which having the recognition of the microorganism by the primary antigenic exposure. Whenever the primary antigenic exposure is formed, the adaptive immune system form the lag phase. In lag phase, they recognize the organism and produce the antibody. After the primary antigenic exposure, the IgM, that is immunoglobulin M, it start to peak, and then in serum the condition is steady, and then suddenly is fall down and showing the negative phase, which is the primary immune response. In secondary antigenic exposure, we can see the negative phase also. So negative phase means a phase of the lower resistance that may follow the reinjection of the foreign antigen in active immunization, or any process that stop, prevent or reduce the frequency, rate or extent of the immune response. So the immunological reaction of any organism to an immunologic stimulus. So after negative phase, suddenly the IgM is high titer. So immunoglobulin G antibody is start to get response against those secondary immune response against the microorganism, that is secondary immune response. In primary immune response, we see the immunoglobulin M is formed, that is antibody M, where in secondary immune response we see the level of IgG, that is immunoglobulin G, that is antibody G, is producing highly, which is initiate the response against the microorganism, that is secondary immune response. So basically, when the same antigenic exposure occurs subsequently, this event which is take place is following. So basically, secondary immune response means the same antigen which is exposure in first time also exposed at the same antigen exposed occurs subsequently, which is called as secondary immune response, and we get immunity against those microorganisms by the producing the antibodies. So some events are take place in secondary immune response also, that is latent phase, negative phase and antibody struck. So latent phase means recognition of foreign antibody, so there are no production of antibody produced, but activate the B cells. So this latent phase maybe can be live 2 or 3 days or often longer. Negative phase means this phase towards the resistance may follow the injection of foreign antigen in active immunization. An antibody shrug means production of antibody, actual fight against the microorganism by producing the antibody, specific antibodies against that antigen and pathogen. So that is secondary immune response. So that is the active immunity. So what is meant by the passive immunity? So passive immunity is defined as the resistance that is transferred passively to the host in the ready-made form without active participation of the host immune system. So passive immunity can be defined as the resistance. Okay, so this is the resistance which is transferred passively from the outer side and which is not formed in the body of organism or host immune system. It is ready-madely put in the body of organism, that is passive immunity. So passive immunity also having the two types, that is naturally and artificially. So natural passive immunity involve the immunoglobulin G antibody transfer from the mother to fetus across the placenta. So this natural passive immunity involve the antibody G antibody transfer from the mother to fetus across the placenta, where artificial passive immunity develop following the ready-made transfer of the commercial prepared immunoglobulin, that is antibodies prepared artificially. It is ingested in the body of organism, that is rabies immunoglobulin. These antibodies are useful for against the rabies virus. So these are put in the body of organism or ingested in the body of organism by the outer side. So this passive immunity is also useful for quite useful for the fight against the viruses, bacteria or microorganism. So let's see some roles of the passive immunity. So immunodeficient individual. So in immunodeficient individuals, passive immunity having the key role to fight against the microorganism, because the immunodeficient individual cannot produce the host immune apparatus that are not effective. So this immunodeficient individual doesn't carry the active immunity or natural immunity in their body system, so that they for fight against the microorganism, they having the passive immunity. So passive immunity is quite useful for the immunodeficient individual. Then post exposure of the prophylaxis, when the immediate effect is warranted. So sometimes some microorganisms showing the some post exposure prophylaxis or some infection, high infection in the body of organism, so the passive immunity can be triggered that response against the microorganism and eliminate them from the body of organism. So passive immunity also develop faster, because passive immunity will inject from the outside, which consists the specific antibody against the specific microorganism, so that they can develop the immunity fasterly. And there is a no lag phase, that is recognition of microorganism, then production of the B cells, then differentiation of plasma cells and negative phase. So there is no lag phase, no negative phase. They start to fight against the microorganism immediately after the injection of the body of organism. So there is no immunological memory. We know that the active immunity having carry the immunological memory, but passive immunity doesn't carry the immunological memory, as the memory cells are not involved, because in passive immunity memory cells are not involved. So we know that the memory cells are the T cells and B cells. So passive immunity doesn't carry the memory cells, so that they doesn't carry the immunological memory. Also they doesn't carry the boosters, which are not effective in the passive immunity. So let's see some difference in primary and secondary immune response. So primary immune response, when immune response against the primary antigenic challenge, whenever the organism or human get first exposure of organism, then start to get response against that antigen, which is called as primary immune response. Secondary immune response means the immune response against the subsequent antigenic challenge. Secondary immune response means same antigen get invented in body of organism in second time, and immunity start to get response against those antigenic challenge. Primary response is slow, sluggish, appear in late and short-lived, because they have to recognize the organism, then start to produce the B cells, then antibody. So they are a slow, sluggish process, and it is short-lived. But secondary response is prompt, powerful and prolonged, long lasting. primary immune response having the lag period, which is longer than the 4 to 7 days, where secondary immune response is absent or short because they having the recognition or memory for the recognition of microorganism. So there is a no lag phase, or it is short, it is 1 to 3 days. So primary immune response doesn't carry the negative phase, but secondary immune response having the negative phase may occur. So in primary immune response, antibody produced in low titer is of immunoglobulin M type. So we know that in primary immune response, immunoglobulin M type is produced in low titer, and antibody are more specific but less avid. So these antibodies are more specific towards the microorganism but less avid, where the antibody produced in high titer, we know that in secondary immune response the antibody produced in high production, which is type IgG, immunoglobulin G type, is produced in secondary immune response, which is less specific, produce the less specific antibodies but more avid, so having the more time to get interact with the antigen, where in primary immune response antibody produced cells, that is naive B cells, that is new B cells are produced in the primary immune response, where in secondary immune response antibody producing cells like memory B cells produce the antibodies, because memory cells having the memory to recognize the microorganism, and they suddenly start to respond against those microorganisms by producing the specific antibodies. So that is memory B cells produce the antibodies in secondary immune response. So in primary immune response, both T dependent and T independent antigen are processed. So in primary immune response, T dependent and T independent antigen are processed, which is fight against the microorganism. But in secondary immune response, only T dependent antigens are processed. So these are the difference in primary and secondary response. So in this lecture we are talking about the what is meant by the active immunity and what is meant by the passive immunity, and first primary immune response and secondary immune response and their differences. I hope you understand this lecture. Thank you.